neurons are cells which are specialized in the transmission of electrical signals called nerve impulses. They have a cell body. They have a cell body. If you look over here, look, they have a cell body. That cell body is going to be responsible for having your nucleus, mitochondria, rough endoplasmic reticulum, ribosomes, all of these other organelles that are needed for making your neurotransmitter, right? Neurons have many thin extensions from their cell body. So from the cell body, they have many thin extensions, right? They are called dendrites. Can you see these small branches? These are your dendrites. So these are called dendrites and they connect to the neighboring nerve cell. The most distinctive feature of all nerve cells is the nerve fiber, which are extremely long and thin. So we have the nerve fiber like this over here. This is also the nerve fiber. And fibers that carry impulses away from the nerve body are called axons. So this is the cell, right? So those fibers that are that is going to be carrying it away from the nucleus, away from the cell body, that is going to be known as an axon. So this is the cell body. So whichever one is going to be taking it away from the cell body, that's going to be known as the axon, right? Fibers that transmit impulses towards the cell body are known as dendrons. So whichever one is taking it towards the cell body, that is going to be known as the dendron. So this is the dendron because it's taking the impulse towards the cell body. This is the axon. It's taking it away from the cell body. This is all axon because all of it is going away from the cell body, right? And short relay or connector neurons, this is a connector neuron, are found in the central nervous system and they connect motor and sensory neuron. So this relay neuron is going to be present between the motor and sensory neuron, right? And we also call them bipolar neurons because they have both fibers, the axon as well as the dendron fibers. Now, when we go ahead and we talk about it, we know about a nerve impulse because that's what the entire topic is about. It's about nerve impulse, right? So the current model of a nerve impulse is of minute electrical event that is produced by charge differences between the outside and the inside of the neuron membrane. And when I go over this, you will see this in more detail. You're going to understand this in more detail, right? Basically, there is a change in the charge between the outside and the inside of the membrane. That is basically because of movement of ions, because of special pumps, because of special pores, and the movement of ions causes a difference in the outside, the charge in the outside, in the inside of the nerve membrane, and that is what allows the electrical impulse to be transmitted. Now, when we talk about our nerve fibers, so our nerve fibers, some of our nerve fibers, not all of them, are myelinated. What is myelinated? What do we mean by myelinated? We have a special type of cell around that is wrapped itself around the nerve fiber. That is known as Schwann cell. So Schwann cell is a specialized kind of cell that will wrap itself around the nerve fiber, wrapping itself multiple times, and it will end up forming this fatty layer. That fatty layer is going to be known as the myelin sheath, right? And what happens is that this is not a continuous layer, right? There are going to be gaps present between the myelin sheath. The gaps between the myelin sheath are known as nodes of Renvia, right? Now, the myelin sheath is important for two reasons. First of all, it protects the nerve from damage, and secondly, it speeds up the electrical impulse transmission. So the electrical impulses are going to be transmitted much faster in myelinated nerves as compared to non-myelinated nerves, right? So, when we talk about that, the role of nerve cells is to carry electrical impulses rapidly from one area of the body to another. The speed at which impulses can be carried depend upon generally two things. The diameter of the nerve fiber. The thicker the fiber is, the more rapidly the nerve impulse will travel. The second thing is presence or absence of this, the myelin sheath. Myelinated nerve fibers can carry impulses much faster than the non-myelinated nerve fibers. So invertebrates, for example, they do not have myelin sheaths on their nerve fibers, right? On the other hand, a lot of your vertebrates actually do have myelinated fibers. So myelinated fibers actually make it easier for the electrical impulse to be transmitted. Now, I told you that the entire process or the entire concept of electrical impulse transmission is due to the change in electrical impulse between the cell membrane and the outside of the cell membrane and the inside of the cell membrane. And we can actually go ahead and evaluate, right? So, we can evaluate them by using electrodes. So, what we can go ahead and do that is that we can this experiment was done in which they took a very fine glass micro electrode and they inserted it into the axon, right? Another electrode recorded the electrode potential from the outside of the axon. So they recorded the electrode potential inside of the axon and they also recorded the electrode potential on the outside of the axon, right? So what happened? That an electrode made from a glass pipette pulled to a sharp tip is filled with an electrically conducting solution, right? And then you connect it to by a wire. And after that, you will insert it both inside the axon and outside the axon. Two electrodes, as you can see over here, one inside and one outside the axon are present, and they detect the difference in voltage in an unstimulated neuron. So in an unstimulated neuron, they record what is the difference in the voltage between outside and inside of the axon. Now, the small difference is then going to be amplified by the amplifier and it will show you the results on the screen. So the difference between outside and inside of the axon is minus 70 millivolts. This is known as resting nerve potential. So the resting charge or the resting nerve potential of our nerve is minus 70 millivolts, right? And that is minus 70 millivolts, okay, and that is going to be true for all nerve membranes. So the resting potential that we have is minus 70 millivolts. Now the questions comes is that how is this how is this important for us? This is important for us because this helps us understand how the electrical impulses are actually passing through, right? So that is going to happen. How our electrical impulse is going to be passed through? The nerve impulses depend upon the concentration of sodium ions, potassium ions, and other charged particles, but mostly sodium ions and potassium ions, right? The membrane of an axon is partially permeable, and its difference in permeability of this membrane to positively charged sodium and potassium ions that give it the special conducting properties. So the axon membrane is relatively impermeable to sodium ions. So sodium ions cannot directly diffuse through the membrane, right? On the other hand, potassium ions can freely cross the axon membrane. And this is going to be important for us. First thing first, what is the resting potential that I just told you? Resting potential, so if an axon is not conducting a nerve impulse, we will say that it is resting, right? The concentration of the ions outside the nerve fiber is greater than the concentration in the cytoplasm of the axon. So the ions are greater outside the axon as compared to the inside. The gradient is created by a very active sodium potassium ion pump. This sodium potassium ion pump is going to be responsible for pushing the sodium ions out and the potassium ions in. So as a result, there is going to be high sodium concentration outside the cell and low sodium concentration inside the cell. And this pump basically has an enzyme that is known as sodium potassium ATPase, and it basically uses ATP to move the sodium ions out of the exon and move the potassium ions into the exons. So what's going to happen? Outside the exon we have a lot of sodium but less potassium. Inside the exon I have very less sodium but I have a lot of potassium, right? The membrane is impermeable to sodium ions. Sodium ions cannot diffuse through the membrane, right? At the same time, potassium ions are actively moved into the exon by the pump. However, because the exon is permeable to potassium ions, so the potassium ions can diffuse through the membrane, right? So what's going to happen, that the sodium ions cannot, they are being pumped out of the membrane and they cannot come back in. Potassium ions are being moved in, but they can also move out. They can also diffuse out down the concentration gradient. So what happens that as a result, the inside of the cell is slightly negative compared to the outside. So the outside has more of a positive charge because it has a lot of sodium. At the same time, since the potassium ions are able to diffuse out, so there is a lot of positive ions outside, not as many positive ions inside. So that creates a difference in the potential, right? So the inside is always slightly negative. So the term we use is polarized, that the membrane is always polarized, right? And the potential difference compared to the outside and inside of the cell is going to be minus 70 millivolts. Now, for our actual nerve impulse to be conducted, we have something known as the action potential. What is the action potential? When an impulse travels across the axon, there's always going to be a change in the permeability of the cell membrane to sodium ions, right? So what's going to happen, that there's going to be change in permeability of the sodium ions. So when a neuron is going to be stimulated, so it can be stimulated by some stimulus like light, sound, touch, taste, smell, pressure, pain, whatever, right? Or maybe a neurotransmitter came and attached to the nerve. Whatever happened, the nerve got stimulated. The axon membrane will show a sudden and dramatic increase in its permeability to sodium ions. First, when it was resting, there was no permeability. Sodium ions were not permeable. But when the nerve will get active, special sodium ion channels, or we call them sodium gates, the sodium gates will open up, allowing the sodium ions to diffuse into the nerve, to diffuse inside the cytoplasm of the axon down the concentration and the electrochemical gradient. Now, since the ions, sodium ions, are suddenly going to all be rushing inside the membrane. So inside of the membrane or inside of the nerve is negative. When they're all going to start entering into the nerve is suddenly going to become positive because a lot of the positive ions have rushed inside the nerve. This is going to be known as depolarization. So when the inside of the nerve was negative, we called it polarized. So this is depolarization because we're doing depolarizing it, we're making it more positive. And this is happening because of the voltage gated sodium channels. Voltage gated sodium channels will open, all of the sodium ions will diffuse inside the membrane, and the inside of the membrane will suddenly become positive and the potential will become plus 40. So from minus 70 it will become plus 40 millivolts. So you will see that it's suddenly becoming positive. That is depolarization, right? This is known as the action potential. Now, after the depolarization, this only happens for about 1 millisecond. The sodium ion channels will close again. The voltage graded channels will close again. And what's going to happen, that the sodium pump is going to again remove all of the sodium ions back outside, right? So also, so that's what's going to happen. So all of the sodium ions that had rushed in during depolarization, all of the sodium ions that had come in during depolarization, they are basically going to move out, they're going to be pushed out, right? And who is going to do that? The sodium potassium pump, the same one that was doing it before. That same sodium potassium pump is going to the same sodium potassium pump is basically going to go ahead and basically going to go ahead and give you that result, right? So what is going to happen? So what is basically going to happen is that the sodium potassium pump is going to push all of that outside. It's going to push all of those sodium ions outside, and as it's going to push all of the sodium ions outside, what's going to happen, that the charge inside the cell membrane or inside the membrane of the nerve will start to slowly come back to negative again. At the same time, the permeability of the membrane to potassium ion will also be increased. So the permeability of the membrane to potassium ion is also going to be increased, right? So this is, why does this happen? Why does the membrane become even more permeable to potassium ions? Because the voltage dependent potassium ion channels will open. Voltage dependent potassium ion channels are going to go ahead and they're going to open, and as they're going to open, so the potassium ions are also going to rush out, or they're going to move out of the nerve, and the sodium ions are also going to move out of the nerve. As a result, what's going to happen, that the nerve potential is going to fall down. It's going to become negative. It's going to become so negative, it's actually going to go below -70, right? It's actually going to fall back down to down below -70, like this. If this is your graph, so it's going to come down below, this is -70, so it's actually going to come down below -70, right? So what is going to end up happening, that the inside of the axon is going to become negative again, right? This is going to be known as repolarization. The nerve is becoming repolarized, it's gaining the polarization again, and after that slowly the potential will come back to -70. So this is how the process actually takes place. Now, the events of action potential depend upon something known as a threshold. Threshold is like the minimum requirement. If there is enough stimulus, if you receive a strong enough stimulus to cause a potential difference that meets the threshold, so an action potential will take place. If the stimulus is not strong enough to meet the threshold, no action potential is going to be generated, right? Now, the recovery time of an axon is known as refractory period. So once the nerve has been, once the nerve has been activated, it is going to be, it's going to recover for a period of time. That recovery period is known as refractory period, right? This is the time it takes for an area of axon to recover after an action potential. There are two types of refractory periods that we have: absolute refractory period and relative refractory period. Absolute refractory period is the period during which no action potential will be generated, right? Sodium channel ions are completely blocked and the resting potential has not been brought back to normal. No action potential can be generated at all. That is absolute. Relative action potential is refractory period is the time in which action potential can be generated only if the stimulus is strong enough. So if the stimulus is that great, if the stimulus is that strong, action potential can be generated. That is going to be known as relative refractory period. And during this time, the voltage dependent potassium ion channels are open. right? So that is why it can go ahead and it can happen. Any confusion so far? You want to ask anything about the topic?
No, miss, I got it.
Okay, all right. Now, when we go ahead and we basically talk about our process of, your conduction, right? So we have something known as saltatory conduction. What is saltatory conduction? So in myelinated neurons, the situation is much more complex. Ions can only pass in and out of the axon freely at the nodes of Ranvier. This means that action potential can only occur at the nodes, and so they appear to jump from one node to the next. This speeds up the transmission of the movement of ions associated with the action potential, right? And this is what allows the myelinated fiber to give you a much faster response. This is why with myelinated fibers we see such a fast response, because of the fact that your action potential can only occur at the nodes of Ranvier. So the action potential is going to jump from one node to the other, right? So that is going to that is basically known as saltatory conduction. Then we need to talk about something known as synapse. Synapse is something that connects two neurons to each other, right? So it is something that connects your two neurons to each other. So neurons must be able to communicate with each other. Receptors must pass their information to the sensory neurons, and then the sensory neurons must take the information to the central nervous system, and information needs to pass around the central nervous system, needs to come back to the motor neurons, needs to come to the effector organs. So all of this we need our synapses that will allow the neurons to communicate with each other. So every nerve in the central nervous system is covered with synaptic knobs, right? And when we go ahead and talk about it, so how does the process actually happen? This is our presynaptic terminal. This is the postsynaptic terminal. This is the presynaptic membrane. This one is the presynaptic membrane, and this is the postsynaptic membrane. So the functioning of the synapses depends upon the movement of calcium ions. The arrival of an impulse, the impulse is coming from here, down there, arrival of impulse at the synaptic knob increases the permeability of the membrane. Which membrane? This one, presynaptic membrane. Increases the permeability of the presynaptic membrane to calcium ions, because the calcium ion channel will open. So there are calcium ions channel on this membrane, presynaptic membrane. They will open. The calcium ions are going to move from the outside to the inside of the membrane down the concentration gradient through diffusion. Now, why do calcium ions move in? The calcium ions cause the synaptic vesicles. There are certain vesicles called synaptic vesicles. Synaptic vesicles contain the neurotransmitter, the chemical that allows the electrical impulse to pass through. So the neurotransmitter is inside the synaptic vesicles. So the calcium ions is going to cause the synaptic vesicles to fuse with the presynaptic membrane. And what's going to happen? As the vesicle is going to fuse, they're going to release the transmitter into the space. This space is known as synaptic cleft. So by exocytosis, the neurotransmitter will be released into the synaptic cleft. Over here, the neurotransmitter is going to pass on to the postsynaptic membrane. The postsynaptic membrane has these receptor sites for sodium channels. Okay. So when the neurotransmitter is going to bind to these receptors, the sodium channels will open. So what's going to happen? The sodium ions are going to pass on from this nerve to the next nerve, and as a result there is going to be depolarization and the electrical impulse will pass from one nerve to the other, right? And the nerve impulse that is generated in the postsynaptic neuron, we call it excitatory postsynaptic potential. So as the sodium ions will diffuse into the postsynaptic neuron, excitatory postsynaptic potential is going to be set up. So if there is enough excitatory postsynaptic potential, what's going to happen? The charge in the postsynaptic cell is going to become more negative. It's going to become greater than the threshold level, and an action potential is going to be generated and the electrical impulse is going to be transmitted. Now, in some cases, not all neurotransmitters are stimulating. Some neurotransmitters are also inhibitory, right? So if they're inhibitory, then the action potential that's going to be generated is going to be inhibitory postsynaptic potential. So there's going to be less action potential that's going to pass through, and as a result there is going to be less stimulus in the postsynaptic membrane. This is how some of the painkillers work in this way, by blocking these for example receptors, right? Or they work in some way where they actually cause the release of a neurotransmitter that has an inhibitory effect. So what's going to happen? That the neurotransmitter is going to inhibit. It's going to do an inhibitory postsynaptic potential. So as a result, the nerve, the painful nerve impulse will not pass through. So that can be a way in which we can actually prevent pain from happening. So a neurotransmitter can be excitatory or inhibitory. Once the transmitter has done its job, it needs to be destroyed. So there are enzymes in the synaptic cleft. They destroy the neurotransmitter, and the neurotransmitter is broken down into smaller pieces. And after that, it is taken back into the into the presynaptic terminal. Any confusion?
No, miss.
Okay, just give me 1 second. क्या हुआ? मुझे नहीं पता. मुझे नहीं पता. वो रो रही थी बहुत. मुझे नहीं पता कहाँ है. तुम कहाँ चली गई? देखो मुझे नहीं पता वो कहाँ है. जाओ देखो वो सोई नहीं है. मुझे सुनाना था 5 बजे तक. जाओ दे दो मुझे. अगर वो बहुत रो रही है तो मैं देख लूँगी. Right. So after that, we have to talk about what are the different neurotransmitter substances that we have, right? One common neurotransmitter found in majority of the synapses in humans is acetylcholine. Right? Acetylcholine, the chemical is actually made up in the synaptic knob using ATP produced in many mitochondria present, right? Nerves that use acetylcholine as their transmitter are known as cholinergic nerves. So the cholinergic nerves are the one that actually use acetylcholine as their neurotransmitter. So the acetylcholine, once it has traveled across the synaptic cleft, it is going to bind to the acetylcholine receptors. The sodium channels will open, the sodium ions will diffuse through, and your excitatory postsynaptic potential is going to be set up. After that, once the acetylcholine has done its job, it's going to be broken down by an enzyme known as acetylcholine esterase. Right, this enzyme is embedded in the postsynaptic membrane next to the acetylcholine receptors. Once the neurotransmitter has attached to the receptor and initiated a response, it is rapidly broken down to give us acetyl. So the acetylcholine will be broken down to give us acetyl and choline. It's going to be hydrolyzed by this enzyme, acetylcholine esterase. After that, the components are going to rapidly diffuse back into the presynaptic membrane, and then the presynaptic membrane, the acetyl and choline can again be combined to make more acetylcholine. Then we also have some other neurotransmitters. We have noradrenaline, right? And the nerves that use noradrenaline, we call them adrenergic nerves, right? So it also goes ahead and it works in a similar way. So these are the different neurotransmitters that we have. Now, how a lot of the drugs that we use in our for our management of nervous system, they basically work by working on the synapse, right? For example, we can have drugs that can make the response stronger. So they can either increase the amount of neurotransmitter that is being made. So more neurotransmitter is going to be made. You can go ahead and do that, right? Or they can increase the release of the neurotransmitter from the vesicles at the presynaptic membrane. So if there is going to be more neurotransmitter, right, being released, so the there is going to be more neurotransmitter binding to the receptor, so more action potential will be generated in the postsynaptic neuron. Then it can bind to the postsynaptic receptors and activate them. So I can also give drugs that are similar to the neurotransmitter. So they will bind to the neurotransmitter receptor, they will activate the neurotransmitter receptor, and as a result, the channels will open and the ions will pass through. It can prevent degradation of the neurotransmitter so that the neurotransmitter is not broken down. So if the neurotransmitter is not going to be broken down, it's going to stay attached to the receptors and the action potential will be generated. Then we can have drugs that decrease the response. So either they can block the synthesis of the neurotransmitter, so neurotransmitter is not being made at all. Then it causes the neurotransmitter to leak from the vesicles and be destroyed by the enzyme. So instead of the neurotransmitter staying inside the vesicle, it will leak out and then the enzyme will destroy it. Prevents the release of neurotransmitter, so the neurotransmitter will not be released, so it will not bind to the receptor, and blocks the receptors and prevents the neurotransmitter binding. So you can also block this receptor. If the receptor is blocked, then even if the neurotransmitter is present, it will not be able to bind to the receptor. So we can do both things. We can increase the response and we can decrease the response. So there are different drugs that we have available, right? For example, we have nicotine. Nicotine is an addictive drug that is often found in tobacco. How does it work? It mimics the effect, or it copies the effect of acetylcholine, and it binds to the acetylcholine receptors in the postsynaptic membrane. These receptors are known as nicotinic receptors. Okay, so it will bind to the acetylcholine receptors, nicotinic receptors in the postsynaptic membrane. It triggers an action potential in the postsynaptic nerve, right? So what's going to happen, that it triggers an action potential, so the heartbeat rate is going to increase, the blood pressure is going to increase, all of these are going to increase. That's how nicotine work, by copying the effect of acetylcholine, right? So that it will bind to the receptor, and as a result, what's going to happen, that there's going to be more stimulation, so the heart rate is going to increase, blood pressure is going to increase. Nicotine not only increases acetylcholine, but it also increases one another neurotransmitter that is known as dopamine. Dopamine is a neurotransmitter that is associated with pleasure, with happiness. So at low doses, it has a stimulating effect. It stimulates the release of dopamine, so the person feels happy. But at high doses, nicotine can actually block this receptor, acetylcholine receptors, and it can actually kill. And nicotine is a highly addictive drug. This is why, because it releases dopamine, so the person feels happy when they have nicotine, so they become addicted to it, right? Then another drug we use is lidocaine. Lidocaine is an anesthetic, it's a painkiller, right? It is generally used by dentists, and lidocaine actually blocks the voltage gated sodium channels, right? So the voltage gated sodium channels over here in the postsynaptic neuron, they will be blocked. So what's going to happen, that this sodium ions will not be able to move, and since they will not be able to move, the action potential will not take place and we will not feel any pain. right? Then we can have something known as cobra venom, which is also known as alpha cobra toxin. This is a substance made from several species of the snake that is known as cobra. It is toxic and often fatal in snake bites. How does it work? It binds reversibly to acetylcholine receptors. So the acetylcholine receptors, it will bind irreversibly to it, right? As a result, the nerve impulse will not be able to pass from one synapse to the another. So the muscles are not going to be able to contract. Eventually the heart will not be able to beat, the breathing muscles are not going to be working, and slowly the person is going to become paralyzed. And as the toxin is going to reach the respiratory system and the muscles of respiratory are not able to conduct impulses, they will become paralyzed and the person is going to die. Any confusion in this?
No, miss, I got it.
All right. Okay, Manur, we'll go ahead and stop it over here for today. We are almost done with the topic. We have just a little bit more left to cover.